metabolism that goes on in our red blood cells. This is talking to or pertaining to after the cell has matured. Okay, the metabolism that goes on in the precursor cells eventually winds up at this. Okay, but without a nucleus, a lot of the normal cell pathways are not available. There is no protein production in an erythrocyte. There is no nothing in an erythrocyte. All you have is basically just a little sack of hemoglobin. But it still has to go through it still has to have energy. It still has to be able to metabolize glucose. We can't starve the poor little things to death. So while we still have a little bit of aerobic glycolysis, but most of our glycolysis in our erythrocyte, and glycolysis is just the breakdown of glucose to form ATP, and ATP is energy, basically. All right. The Ebden-Meyerhof pathway is our anaerobic glucose metabolism. Within this, we also have the Rappaport-Lubering pathway, which generates 2,3-BPG, and we'll talk a lot more about that tomorrow. And hemoglobin reductase pathway maintains the iron in the reduced states. We'll get more into it. It'll make more sense as I finish the lecture. Okay. Our aerobic glycolysis is the hexophosphate shunt. This also is not just doing glucose, but it generates NADPH, which helps protect the cell. Nucleotide metabolism must be maintained to preserve energy for the cell. This is one of those chicken and the egg things. This is a list of what we do. Now we're going to go through each one of them. They'll make sense as we go through them, okay? The thing you need to remember for these, it will be on the test. You need to know the pathway, the enzyme, because this is going to become very important later on in lecture, and what it makes. So you will, so you will need to be able to match these up. This is Rappaport-Lubering, this is hexose monophosphate shunt, and this is the Ebden-Meyerhof pathway. Okay? And especially PK and G6PD are huge when we are talking a little bit later about hemolytic anemias. So you need to know those pathways, those enzymes, those products.
What's the EM stand for in pathway?
Ebden-Meyerhof. The names are on the previous slide. That's why the names are on the slide, so I could make the chart with the abbreviations. The structure of hemoglobin, just the hemoglobin molecule itself. It is a conjugated protein molecule, which means that it's a protein hooked onto something else. There are 4 heme groups, and we'll talk more about what's in each of those, and 2 pair of unlike polypeptide chains. So if there are 2 pair of chains, how many chains are there in a hemoglobin molecule? If I have 2 pair of shoes, how many shoes do I have? 4. So if I have 2 pairs of globin chains, how many globin chains are in my hemoglobin molecule?
2. You just asked chains. You didn't say.
He only wears
How
one
many
shoe at
chains
a
are
time.
there? Yeah.
Yeah. No, there's 2 pair. So a pair is 2. A pair is 2. Okay, there are 2 pair of chains. Okay. The heme is a protoporphyrin ring. Okay. You can look up in your book, and it's got a lovely picture of it. That's nice. It's exciting. It's your basic carbon, hydrogen, and nitrogen ring. It's just like any other organic molecule you're going to find in your body. I am never going to ask you the structure of the protoporphyrin ring, so don't spend a lot of time on it. Okay. It has an iron attached, so it's basically, it's a ring, small thingies coming off the outside, and in the middle of the ring, we plop an iron. It has to be ferrous iron, which has a valence of 2. If we get ferric iron, which is a valence of 3, we have a problem. So we have to have ferrous iron. The globin, there's 2 pairs of polypeptide chains, and we'll talk a lot more about those in a little bit. It has somewhere between 141 and 146 amino acids. Yay, hooray, hurrah. I will never ask you how many amino acids are in a chain. So here's a picture of our hemoglobin molecule, or of our heme. This is a heme group, a single heme group. And we've got the A, B, C, and D quadrants. Basically, they're pentamers. That's nice and exciting. Big thing, nitrogen hooks to iron in the middle, okay? It can carry up to 4 molecules of oxygen because the oxygen comes in and binds to this ferrous iron. The fact is, most importantly, it reversibly binds to the iron. It's not very monogamous. It comes in there, but as soon as something better comes along, it takes off. So it just hooks onto the iron to get transported to where it needs to get. Biosynthesis of heme, in other words, where is it made? It occurs in the mitochondria of the RBC precursors. Hemoglobin is not made once the cell is grown up. Once you have an erythrocyte, there is no longer any production of hemoglobin. What you get before it spits out the nucleus is all you're going to get. You can't put more in, you can take it out, but you can't put more in. You need to remember this first step, yes you do. Glycine and succinyl-CoA, and that's how we refer to it, glycine and succinyl-CoA, form ALA. I will not ask you to remember the tricarboxylic acid cycle, but you need to know that glycine and succinyl-CoA condense to form ALA. And I won't ask you to spell aminolevulinic acid either. ALA will be on the test. The reason this is important is because this is the rate limiting step in the reaction. And if you were really bad in chemistry, this has probably got you guys all freaked out. Chill. A couple of slides later, we move on to biology back where we need to be. The rate limiting step in heme production, what happens is once you've got that completed molecule, the presence of that completed molecule inhibits this TCA cycle. So once I have enough heme, it keeps me from making more. Does that make sense? Okay. That once I've got my final product, it goes in there and basically I've got my final product and everything stops. They stop putting more on the production line because I'm full. So once the heme product is there, it actually inhibits that glycine and succinyl-CoA condensation. You don't make ALA and it doesn't go on down the steps. I do not make you learn all of the steps of hemoglobin production because you got porphobilin and coproporpho and uroporpho. There's 1,000 steps in it, and yes, I can sit down and write it out if I have to. But you don't have to. If it makes you happy and you just like learning obscure, weird stuff, go right ahead. Or if you're a specialist in hematology, you better learn it. Other than that, just know the first step and the last step is heme, and there's a bunches of steps in between. Okay. I don't make you know the full breakdown. The final step, the very last thing, once the heme ring is made, we hook iron with the protoporphyrin 9 ring, and that's what we make in this whole thing. Okay. Iron plus protoporphyrin 9 is heme. And then we attach one of these heme groups to one globin chain, and then hook. Let's start that again. We attach one heme group to a globin chain, and then we hook 4 of those globin chains together to make one hemoglobin molecule. How do we and where do we make globin? Well, it is made within the cytoplasm of the RBC, and it's like any other protein production in your body. It's just a string of amino acids, which is made by taking DNA, making transcript RNA, messenger RNA, put it all together, life is happy. Alpha and non-alpha chains combine to form a dimer. You will need to know which are the alpha and which are the non-alpha chains. The alpha chains are alpha and zeta, A and Z, beginning and the end of the English alphabet. I know omega is the end of Greek alphabet. Alpha and zeta, so A and Z are our alpha chains. These are controlled by a gene on chromosome 16. Do we gotta know that? Did I just ask you? Did I just tell you? The answer is yes, you have to know that. Okay. Chromosome 11 controls our non-alpha chains. Gamma, beta, delta, and epsilon. The ones that have little stars on them, those are only formed in the early stages of hemoglobin production in the fetus. These are not found in anything after birth. They aren't even going to be any living human who has zeta and epsilon chains is not going to be mature enough to survive in the world. Okay, those chains are not adequate for oxygen transfer. Two of the dimers are going to combine to form a tetramer. So basically what we get is we get two alpha chains, because zeta's only in babies. So two alpha chains and two of something else. And depending on what the something else it hooks to, gives us what kind of hemoglobin we have. We'll go through hemoglobinopathies and a ton of that. So we have two dimers, which is an alpha and a non-alpha, hooked together to make a tetramer, which means 4. And it's regulated by the rate of DNA transcription to messenger RNA, just like any other protein production. Do you need to know the rate limiting step for globin synthesis? Yes, you do. So you need to know the rate limiting step for both heme production and for globin synthesis. Structurally normal hemoglobins, okay? When we talk about normal hemoglobins, we got functionally normal hemoglobins and structurally normal hemoglobins and functionally normal hemoglobins. So we got a lot of definitions of normal when we're coming to hemoglobin. In this particular case, we're simply looking at the structure of the molecule. That means that I have a sufficient and appropriate heme group with a proper iron in it, and then we're going to hook it up to chains. In utero, we start with Gower 1. We're going to have zeta and epsilon. And matching question. I'm going to give you the name of the hemoglobin. You got to match it to the proper chains. So Gower 1 is going to give us zeta and epsilon. And if you don't know what the little squiggly symbols are, look on the slide before. So Gower 1 is zeta and epsilon. Gower 2 is alpha and epsilon. Portland is zeta and gamma. Hemoglobin F is alpha and gamma. This hemoglobin F is the primary hemoglobin until children are about 3 months old. Most adults have between about 1 and 2% hemoglobin F. Okay. So this would be part of your adult hemoglobin makeup. But primarily we're going to find this in children till 3 months. Yes?
Do you think you're going to write it out or use the Greek symbols?
Which do you want me to do? I can do both. It's not a problem. I can copy paste where I've got them.
Write it out.
I'll do both. All right, I'll do both. It just was easier on here for me to do this. Okay. Because I knew I'd be talking and telling you what the words were. Okay. Now when we hit the adult hemoglobins, our normal hemoglobin A, which is our adult hemoglobin, okay, is 2 alpha and 2 beta chains. This is what most adult hemoglobin is made of, is hemoglobin A. So I know it's going to be tough, but I'm making you remember that hemoglobin A is adult hemoglobin. If you think you can, hemoglobin F is fetal hemoglobin. I know that's asking a lot. Hemoglobin A2 is also an adult hemoglobin, but it is 2 alpha and 2 delta chains. basically, your body makes lots of alphas, makes lots of betas, makes a few gammas, a few deltas, a few of those other weird ones. As long as you've got normal hemoglobin going on, everything's happy, hunky-dory, you're gonna get your alphas and your betas are gonna hook right up, okay? When you start, and your alphas are gonna hook up with a few of these little, odd ones off to the side every now and then. As we start getting abnormal hemoglobins, we're gonna start seeing a shift in some of that. This is my favorite picture. I don't know why, but it just kind of makes the most sense to me. What you've got this lovely little heme spaceship floating around down there, and it's got this great big protein molecule kind of globbed around it, okay? And there are 4 of these. There are two alpha chains and 2 beta chains. And this is how they'll be arranged. They'll be across from each other like that. I'll never ask you about the cross from each other. But we have 2 alpha and 2 beta chains. What we wind up with is kind of a sort of roundish shape of the molecule. And the little red button in the middle of the heme is the iron. Hemoglobin production, this is what I told you before. This is hemoglobin production is inhibited by the ALA production is inhibited by the completed heme molecule, and the regulation of protein synthesis is regulated by DNA transcription. The hemoglobin production is stimulated by tissue hypoxia. What the heck is tissue hypoxia? What is it? Lack of oxygen. Where does that get measured? In the kidney. What happens when the kidney measures low oxygen? It makes more EPO. And then that goes to the bone marrow and says, make more red cells. The normal hemoglobins, this is just a repeat of what was in an earlier slide. Hemoglobin A is out 2 alpha, 2 beta, and it's about 95% of normal adult hemoglobin. Hemoglobin A2 is alpha and delta, and 2 to 5% somewhere in there. And hemoglobin F is 2 alpha and 2 gamma. It's about 1 or 2% of adult hemoglobin, but who is it the primary hemoglobin for? Fetal, babies. So any baby up to about the age of 3 months, and then from 3 months to adulthood, it's sort of the fetal hemoglobin goes down and the adult hemoglobin goes up. We're going to stop there. I think that's enough. Keep in mind, this is structurally normal hemoglobin. Like I said, we've got 3 or 4 different definitions of normal hemoglobin. So these are those that are structurally normal. I recommend you all take a break. When you come into the lab, I've brought bloods from the hospital, so each of you has your own blood to do a CBC, just one, for your practical, just one. Then I have one slide. It is not cover slip, so please do not wipe it off. Just put it back on the tray with oil on it, and I will clean it later. I have one slide that is labeled normal 1 to normal 12. What I want you all to do is I want all of you to count it. We're going to look for a precision. Does anybody remember what precision is? Yeah, how close together. I want to make sure that you're all kind of in the same ballpark. I think it's normal, but everything I thought is normal, y'all are finding weird stuff on. Once you get that done, if you want to do some more slides from last week, any of them that you didn't get done, last week's tray is still available. I'd like you to do a couple of those, yeah.
Did you know we already did the CBC?
No, we didn't. We did a CBC proficient, we did a competency, but we didn't do the practical. Am I confused? I didn't think so. I know you've done a competency, but I don't think you've done the practical. because the practical's timed and graded. The competency is just a check you did it.
Let's see.
You guys did some CBCs.
Manual white blood cell count.
Yeah, we've done the H&H competency, then we'll do the CBC, then we'll do a slide competency, probably in a couple weeks, and then we'll do the final competency. Or final practical.
Hey Diane,
Yes.
do you have the grades, are the grades up to past 3 still?